Good evening and welcome back to Basic Science Lights the Way. I'm Mike Botchan, as you heard, Dean of the Biological Sciences. We're grateful that you continue to tune in as we share the latest groundbreaking science at UC Berkeley. Tonight, we're going to be talking about breakthroughs in the news, those exciting discoveries that make you sit back and take note. Last month, at an event on campus, Secretary Condoleezza Rice relayed a joke about telling our grandchildren how people used to get their news. Somebody stayed up all night printing it on this piece of paper, and then they threw it at your house in the morning. Now, many people find out important discoveries not long after they're announced. On websites, on social media, through text and apps. The firehouse, or fire hose, I should say, of content can feel like an overwhelming amount. But it's not just the speed in which we get our news, but the pace at which discoveries are happening. In field after field, we're unlocking scientific wonders at a rate never before seen. The researchers tonight will shine a light on our own progress here at UC Berkeley in the fields of physics and biology and what those breakthroughs mean for society. Once we're underway, I hope you will post your questions for any of our speakers in the chat box. We will answer as many of them as we can. Our moderator this evening is no stranger to having her work featured in the news. Barbara Jacak is a professor in the Department of Physics. She is one of the leaders of the nuclear physics community in the United States. Her research focuses on the experimental study of quark-gluon plasma. The plasma is formed in relativistic heavy ion collisions where nuclei are heated to trillions of degrees and quarks are no longer confined in the hadrons. The experiments are done at Brookhaven National Labs, RHIC, and at CERN. She was one of the founding members of the Phoenix experiment at the Relativistic Heavy Ion Collider, that's the RHIC, and was a spokesperson for the experiments from 2007 until 2012. Before joining Phoenix, she was active in the CERN heavy ion program as a member of the Helios and 844 collaborations. She is currently working on the ALICE experiment at CERN, LHC, and the EPIC experiment for the Future Electron Ion Collider. She has been very active in the development of the National Nuclear Physics Program and served on the Long Range Planning Committee, Long Range Planning Group. She is a fellow of the American Academy of Arts and Sciences, the AAAS, a member of the National Academy of Sciences, and a member of the American Philosophical Society. We're delighted to have you here this evening as our moderator. And Barbara, it's now over to you. It's your show.
Thank you, Dean Botchan. I'm excited to be here with you all this evening. Tonight, we're going to hear from some of my distinguished colleagues about some of their most recognized research contributions. From unraveling the mystery of antimatter to the use of CRISPR-Cas9 to cure disease, UC Berkeley scientists are at the forefront of addressing some of the major challenges facing humanity. In essence, science at UC Berkeley not only advances our understanding of the natural world, but also drives innovation and shapes the future of science and technology on a global scale. At the end of each talk, we will try to address all of the questions from the audience, so please be sure to add your questions in the chat. Now it's my pleasure to introduce our first speaker, postdoctoral researcher, Dr. Danielle Hodgkinson. Danielle is a postdoctoral fellow from the University of California, Berkeley, who's found notoriety in the news due to her involvement in the alpha-G experiment, which confirms gravity's effect on antimatter. The alpha experiment at CERN was designed to produce something extremely rare, antihydrogen, and study how its properties compare to those of normal hydrogen. An offshoot experiment, alpha G, has now tested another property of antimatter, its response to gravity. Two UC Berkeley physicists proposed the test a decade ago, and new results show definitively that Earth's gravity does not repel antimatter. Instead, antimatter seems to fall to Earth with the same acceleration as normal matter. Danielle, thank you for joining us this evening. What would you like to share?
Hello, thank you for the introduction. I will share my slides. Okay, so hello everyone. Today I'm going to be talking about our experiment to measure antimatter gravity. And this is work that originally came out of conversations between my two PIs, Joel and Jonathan, shown here many years ago at UC Berkeley. But in 2023, we published this in Nature, and it received widespread media interest. And this is because we attempt to address one of the big outstanding questions in modern physics, and that's the question of how do we exist? We were featured in the New York Times, in The Guardian, and on NSF's Discovery Files podcast, and we are ranked one of Physics World's top 10 breakthroughs of 2023. So to understand this article, let's go back to the beginning. Let's talk about what is antimatter. We're all familiar with the positively charged proton. The antimatter equivalent is the negatively charged antiproton. For the electron, it's a positively charged positron. Antimatter is having the same mass but opposite charge as matter, and every particle has an antiparticle. And when matter and antimatter meet, they annihilate to produce photons. In the reverse reaction, a photon can produce a particle-antiparticle pair. In both of these reactions, annihilation and pair production, equal amounts of matter and antimatter are either being created or destroyed. And we think that during the Big Bang, also equal amounts of matter and antimatter would have been created. And then, as the universe expanded, we expect that these equal amounts would have annihilated. But this is in direct contrast with the observation that our universe today appears to almost entirely consist of matter. We do see small amounts of antimatter, such as in radioactive decay, like the handful of positrons that come from bananas, but it's nowhere near equal to the amount of matter in the universe. So we think that at some point in the early stages of the universe, some process must have created an excess of matter over antimatter. But our current understanding of physics can't explain this. And so at Alpha, we look at the fundamental properties of antimatter atoms, and we compare them to known properties of matter. And we look for small differences between the two that might be a clue towards how our theories need to be revised to explain the matter-dominated universe. And one such property is its gravitational interactions. Now, based on our current model of gravity, Einstein's general theory of relativity, and specifically within that, the weak equivalence principle, we expect that antimatter atoms should fall down in the Earth's gravitational field just like matter atoms. But there do exist theories which predict the opposite, that antimatter actually falls up in the gravitational field of the Earth. And so that's what we test. Actually, in the 60s in Stanford, Witterborn and Fairbank came up with an experiment to measure this. And they wanted to get some positrons inside of a vacuum chamber and drop them to see, do they go down or do they go up under the force of gravity? But they started with electrons, and they got the surprising result that the electrons did not move. And that turned out to be due to charge buildup on the walls of this vacuum chamber. And because the electron has charge, it receives a strong force from that electric field. And that completely overwhelms the force of gravity and makes it impossible to measure using this method. So to get around this, at Alpha, we trap antihydrogen atoms, and they have no charge, they're charge neutral. And so we can detect the gravitational interactions without being overwhelmed by electrostatic forces. And we trapped antihydrogen for the first time in 2010 in Alpha, and it's trapped by combining positrons, which we take from radioactive decay, with antiprotons. And to get the antiprotons, we use the same methods that are used in the Botchan in Berkeley. That is accelerating protons very fast, hitting them into a target, and then some of the products of that hit are antiprotons. And this is the work which led to the antiproton discovery by Chamberlain and Segre in 1955, for which they were later awarded the Nobel Prize. But the one place in the world that you can do this right now is in CERN's antimatter factory. So here in the antimatter factory, we take those antiprotons from those collisions and we slow them down in this outer decelerator ring known as the antiproton decelerator. They're slowed down so that they can be low enough energy to be trapped in our apparatus. Now let's look at the alpha experiment. On the right-hand side here, we have a radioactive source. We're accumulating positrons. We're taking a bunch of positrons and then we're going to bend them around the corner using magnetic fields up into our gravity apparatus. And then we're going to recapture them. And this is the apparatus known as alpha G. For the antiprotons, we're taking those also down our beamline. Those come from the antiproton decelerator. We're going to bend them up around the corner using a magnetic field and then also trap them in our gravity apparatus in a neighboring well. So here's where we want to do our gravity experiments on antihydrogen. So we want to merge these two plasmas to form neutral antihydrogen, which we can then drop under the force of gravity. So we'll send that antiproton plasma into the positrons. And as we do that in a reaction which involves two positrons and one antiproton, one positron carries away the excess energy and the remaining positron forms a bound state antihydrogen. And in this particular way of making antihydrogen, we make it with a speed of typically 70 meters per second. And if we imagine that antihydrogen is pulled down in the gravitational field of the Earth, and we send it up with 70 meters per second of speed, then it would travel hundreds of meters in the air before being turned over due to the gravitational force. But our apparatus is only 5 meters tall. And so effectively, this antihydrogen atom will just shoot off to infinity if we tried to simply drop it in the gravitational field. So what we do to get around that is to make use of the fact that it has a very small magnetic moment. And that means it's ever so slightly magnetic and it feels a force in a magnetic field. And specifically, it's more strongly repelled from a stronger magnetic field. And that means that if we have a magnetic field that increases in all directions away from the minimum, then we can achieve three-dimensional antihydrogen confinement. An axial confinement is provided above and below the trapping region by these upper and lower coils. Let's have a look, though, now what happens just along the axis. So we can see that we have our magnetic confinement coming from our upper and lower coils, but we also have gravity acting here. So in this case, I've assumed that gravity is pulling the antihydrogen down towards the Earth. And so gravity is creating this asymmetry in the total potential. To make the measurement, we're then going to gradually reduce the magnetic field from those upper and lower coils. And as we do that, at some point, the antihydrogen atom will be able to escape. But because in this case, gravity is pulling the antihydrogen atom downwards, it's making it more likely to escape over the lower coil than the upper coil. And so what we're doing here is we're creating a kind of pan balance. We are trying to measure this small gravitational potential, and in order to do that, we're balancing it with a magnetic field. And so let's do the experiment. Now we've got our antihydrogen atoms trapped, and then we're going to gradually reduce the confining field of those upper and lower coils. And as we do that, the antihydrogen atoms are going to gradually escape. And because they're antimatter, they're going to annihilate with the walls of the apparatus. And that's good for us because we can detect those annihilations, and then we can count how many went down versus how many went up. And in this case, we can see that more go down than up, which indicates gravity is pulling those antihydrogen atoms down. The gravitational field of the Earth is pulling them down. And what we did is we ran that at different magnetic field configurations, which we've labeled biases here. And the experimental data is shown in blue. And we found that that data is consistent with a normal gravity simulation, i.e. the fact that antihydrogen falls down in the gravitational field of the Earth, just like hydrogen does. And we also saw that it's inconsistent with repulsive gravity, i.e. we concluded that antihydrogen does not fall up. And via this experiment, we measured the gravitational acceleration of antihydrogen to be this value in units of the gravitational acceleration of hydrogen. So thank you very much for listening, and I'll open the floor to questions.
Thank you, Danielle. That is truly groundbreaking work. It looks like we do have a few questions from the chat for you. Let me start with this one. What would the implications be if it turned out that the antihydrogen had in fact fallen up?
Yeah, so this is a great question. I'm sure there would have been a lot of shocked theorists around the world because this would have been a very unprecedented result. But it would completely revolutionize our understanding of physics. Our theory of gravity would need to be revised. But that would be wonderful because we would have found something which could give us a clue towards how we've ended up in this matter-dominated universe.
Thank you. Here's a question from the chat. I can't imagine the complexity of tuning all of the apparatus to do what you want in a finite time. Can you describe a bit how it's managed? It's a question from Warren Gish.
Yeah, so tuning of lots of different parts of this experiment is a great point. And one of the big aspects of that is tuning our plasmas. So we want to have our plasmas which are cold enough so that when we merge the antiprotons and the positrons, we get enough low energy antihydrogen that we can actually trap it using our very shallow magnetic potential. And the colder we can get that initial distribution, the more trapped antihydrogen we have and the more statistics we have in our gravity measurement. So there's an awful lot of tuning that goes on specifically with making sure we have the coldest plasmas available. But then also, in terms of our magnetic fields, we have to know our magnetic fields to a very high level of precision, because a value of magnetic field of just 10 to the minus 4 Tesla would completely overwhelm the gravitational potential over the length of the trap. And so we have to also know our magnetic fields very well. We have to have good current control on our superconducting magnets. And we have to have lots of techniques where we can actually go in and measure those magnetic fields and know that we have our fields controlled to an extremely high level of precision.
Cool. Deirdre asks what we can do with this knowledge. Can you give us some examples or maybe say something about the beauty and importance of basic science?
I would say what we can do with this specific knowledge is we can start to rule out different theories that may have predicted that antimatter would fall up in the gravitational field of the Earth. So that allows us to no longer concentrate our efforts on things which are experimentally ruled out. But it also tells us, okay, now we can start to push forward towards higher precision in these experiments. And as we do that, we continue to learn more and more and set ever tighter constraints on the theories around antimatter. And therefore, we can search more and more precisely for something that could be a clue towards how our theories might need to be revised to explain the matter-dominated universe.
And we have a question from Mark Coopersmith. What comes next? With these findings, what do you look to test next? And he also asks, how might we someday apply this new knowledge?
So what we would test next in terms of this specific experiment is, as I said, just pushing towards higher precision. And the way that this experiment was originally designed is that initially we would determine, does it fall up or does it fall down? We've done that. And now we want to transfer our antihydrogen atoms to a different part of our apparatus where we know our magnetic fields to a higher degree of precision. So we'll trap our antihydrogen in what we call the up-down trap, which is where we did this experiment. Then we will transfer the antihydrogen atoms to this more precise region known as the precision trap, and we'll do further experiments there. determining, okay, well, now we've seen that they go down. Do they go down at the same rate as hydrogen? And what we can also do is we can try to reduce the energy of those anti-hydrogen atoms. And as we do that, they become more sensitive to the gravitational force. And so we can employ our cooling techniques that we've demonstrated in our other experiment, alpha-2, such as laser cooling and the topic of my PhD thesis, adiabatic cooling of antihydrogen, to reduce those antihydrogen energies and become more sensitive to the gravitational force.
Thank you. We have a follow-on question from Warren Gish. Over what period of time is the plasma collected and then the gravity and diffusion performed?
Yeah, so we actually, we make antihydrogen in a process which we call stacking. And that involves taking one of our positron bunches, sending it up into alpha G, taking one of our antiproton bunches, sending it up, doing the mixing process, which is just combining those two plasmas. We form antihydrogen in a relatively hot distribution, and then we have this shallow magnetic potential. And so it's so hot that most of them just immediately are not trapped, but the coldest ones remain confined in that shallow potential, and we trap a few atoms when we do that. And then what we do is we repeat that. So we keep our antihydrogen trap on, we keep the antihydrogen atoms that are in there, and then we can take another bunch of positrons, and we take another bunch of antiprotons, and we send those up, and then we trap some more antihydrogen. And that's called stacking, making more and more antihydrogen over time. And so as we're doing that, we're building up the statistics that we need to then do a gravity measurement. And so that is done for one magnetic configuration. We collect about 300 atoms, do that gravity experiment, change our magnetic configuration, combine another 300 atoms. So this data was taken over a period of weeks in total to achieve that full graph that we saw in my presentation.
How long does it take to do one magnetic field?
So how many magnetic fields do we have in total? I think around 6 or something, 6 different biases that we call them. So 3 weeks divided by 6, about half a week or something that we spent on each individual bias, but we interleave them, right? So we're not just focusing on one bias and then changing to the next one, we're interleaving different magnetic field configurations to build up the total statistics that we see in that graph.
So it takes days, not hours or minutes.
Yeah.
Great. And another interesting question from Shamin Chowdhury, which is, why would you expect antimatter to fall up rather than down?
So it's not something that we expect to happen, and our motivation is not that we think that that's going to happen. Our motivation is it's something untested in physics, and based on our current understanding of physics, dark matter, dark energy, it's clear that our theory of gravity is not complete. And so even though it's not completely, we don't think that antimatter is going to fall up in the gravitational field of the Earth, we couldn't move forward without testing it. And I think it's very important that we just look and we let nature tell us whether antimatter falls up in the gravitational field of the Earth, and it doesn't. And so we can start to look elsewhere for what might be wrong with our theories involving antimatter.
So we've learned that antimatter is not sneaky, but nature might still be a little bit sneaky.
Yeah.
On this note, he's the director of therapeutic delivery at the Innovative Genomics Institute. He's working to enable widespread clinical use of genome editing, which is currently limited by the difficulty in delivering therapeutic enzymes to the cells in need of correction. To address this need, the Wilson Lab is engineering CRISPR enzymes to create effective methods of administration that rely on targeted delivery to specific cells or tissues or organs. Professor Wilson obtained his BS and his PhD in biochemistry at The Ohio State University. Working as a postdoctoral scholar in Jennifer Doudna's lab, he then studied the roles of protein components in the biogenesis of microRNAs, which are the small molecules that carry out genetic regulation. So, Professor Wilson, please tell us about your research.
Thank you so much for the kind intro. It's a pleasure to be here. So I'm Ross Wilson. My lab is at UC Berkeley, and I'm part of the Innovative Genomics Institute. Today, I'm going to tell you about my work to turn CRISPR into an affordable, safe, and accessible cure for sickle cell disease. So at IGI, I'm the director of therapeutic delivery, and part of our mission at IGI is to maximize the positive real-world impact of CRISPR. And Jennifer founded this institute. You likely know that she was honored with the Nobel Prize in 2020 for her co-discovery of CRISPR-based genome editing. And CRISPR pretty much instantly revolutionized biological research. And it very rapidly moved towards the clinic too, and has made a lot of real-world impact there as well. So before we get to the clinical impacts of CRISPR, I'd like to tell you a little bit about how it works, just in case anyone has not been up to speed yet. CRISPR itself is often compared to a pair of scissors, but it's a very intelligent pair of scissors because it can find one specific location in the human genome and make a cut. And so that's what CRISPR-Cas9 is shown doing here. It's a precise targeted cut in the genome. After that, the cell is sort of responsible for what happens next. Very often, that cut is resolved in a way that changes the sequence and disables the targeted gene. Alternatively, if a DNA template is provided, that can be sort of copy-pasted into the genome, but this is tricky to do. It can be very powerful, though, because you can add new gene functions, like in the case of a CAR T cell to fight cancer, or you could restore a broken gene. There's also a next generation version of CRISPR called base editing, which is more like a pencil with an eraser, where you don't need to break the DNA anymore, and you can go in and erase and replace a single letter to restore function of a gene or turn off a troublemaker gene. So CRISPR rapidly sped towards the clinic. One of the first diseases that was tackled was sickle cell disease. This is because it is really well understood at the genetic level, and there's a huge global burden of sickle cell disease. It shortens lifespans by decades, and there's no real great treatment option out there except for bone marrow transplant from a relative, and not everyone can find a matched relative. So the promise of CRISPR was to enable the patient's own cells to be sent into the lab setting, treated with the CRISPR enzyme to ensure that they could make healthy red blood cells, and then have those blood-generating stem cells return to the patient's own bone marrow to effectively let them be their own donor for these corrected cells. And it worked. By now, over 100 people have been treated with CRISPR-engineered stem cells that can generate healthy red blood cells. And excitingly, in December, the FDA approved the first ever CRISPR therapy. So we now have a CRISPR drug, and this happened very rapidly, going to the clinic just 7 years after the conception of genome editing. So that's a very quick speed for bench to bedside. So as miraculous as this seems, there's also a grueling reality that's associated with these transplant-based CRISPR therapies. Each patient has to undergo cell harvest, which in sickle cell disease can trigger a pain crisis. The patient then has to undergo aggressive chemotherapy, which can lead to infertility. This is necessary to make space in the bone marrow so that the patient can engraft the corrected cells that were engineered in the lab. So once these cells are infused, then the patient has to stay in the hospital for weeks to make sure that they're healthy enough and that their blood production is back up to speed so they can go home. So although CRISPR can certainly work to get the patient to a state where they're generating these cells, the fact that we can't access those bone marrow stem cells inside the body means that this transplant is necessary at the moment. And this is really a delivery challenge. So what my lab works on is finding ways to get those CRISPR enzymes to the cells in need of correction inside the body. And so I'm going to tell you about our work to do that today. And the things that we want to tackle with this improved technology are pretty concrete things that are going to have a real world impact. So we want to expand access by sending these genome editing enzymes into the body with a homing mechanism that lets them find the blood generating stem cells and to update the genome to allow normal red blood cell production. So this should allow deployment in low resource settings, which is very relevant for sickle cell disease, considering how many people have this condition in low resource settings. We also want to minimize risks of toxicity that are associated with the transplantation. Just getting that chemotherapy to wipe out your bone marrow can result in a risk of cancer. And finally, we want to streamline manufacturing of these materials and have something that's really affordable to produce. So ultimately, it's been very clear that CRISPR can cure sickle cell disease, but this therapy cannot be deployed globally without new delivery technology. And that's what my lab is working on. And so we take a molecular engineering approach. We want to expand CRISPR's existing capabilities with new properties for cell-targeted delivery. So I just want to highlight the built-in strengths of the CRISPR enzymes. They can bind to a targeted sequence. This is the specificity we talked about. This part's great. I also compared CRISPR to a pair of scissors that can cut the double-stranded DNA target. This part is great. But CRISPR doesn't have the capacity to discriminate between different cells. And so we want to add a GPS-like function to make sure that the enzymes can navigate to a specific cell type. And once they get there, they also need to get in. So this is where the opener comes in. We need these enzymes to get across cell membranes. So in contrast to traditional small molecule drugs, enzymes can't get across the cell membrane. Cells are very wary about letting foreign proteins in. Very often these can be toxins. So we need to find a gentle but effective way to outsmart these cellular defenses. And the first piece of technology I'm going to tell you about is these short helical peptides that resemble the corkscrew, even at the molecular level. These are cell entry peptides that work really well to get into cells that we want to edit. So my lab recently published a paper on using these cell entry peptides to send CRISPR enzymes into T cells, and we use this to create anti-cancer CAR T cells that work great. So this is our opener, and it seemed to work really well in T cells. Here's our collaborative output trying to show a rendering of what the CRISPR enzymes, blue and purple, what they look like when they're coated with these short stretches of peptide, so little snippets of amino acids that can sort of ferry the CRISPR enzyme into the cells, which are shown in the back here. So we had some success in T cells. We were curious about those blood-generating stem cells that are important for sickle cell cures. And we were very delighted to see that this worked great in these blood-relevant stem cells as well. So using an enzyme called Cas12a, we saw hugely efficient editing in the laboratory setting. And we even saw really nice editing with this base editor, the eraser and pencil that I told you about. And both of these cases were well above this 20% mark, which is sort of the clinical threshold for benefit in sickle cell disease. So this is great. We found an opener. It works for these various enzymes. It works in the blood-generating stem cells that we care about. But we don't have a solution for that targeting. If we really want to go into the body and have this home to the right part of the body, we need the GPS function. So to do that, we imagined strapping the enzyme to a targeting molecule as well as these opener molecules. And the targeting molecule that we have first tested is the antibody. So antibodies are really smart molecules that can recognize specific molecular signatures. And we wanted to harness this power to pull the enzyme towards markers that distinguish these blood-generating stem cells. So in this experiment, we started with a negative control antibody, and under these conditions, we got about 7% of the cells editing. Not great. We wanted to do better. And when we used different antibodies targeting different cell surface markers, all of which can characterize these stem cells we care about, we were able to double the editing. So this suggests that we're en route to really improving the homing capabilities of these CRISPR enzymes. I'll now take a little bit of a detour and tell you about brain targeting, not because it's relevant for sickle cell disease, but just because we have some really exciting proof of concept here that I hope we can translate to the bone marrow targeting example. So in these experiments, we're using a mouse model that actually has a red fluorescent protein that lights up when we edit that region of the genome. So we can turn on a red light for every edit that happens. What I'm comparing here is a cocktail that has the delivery peptide, the opener, and the enzyme. And we also have a version where we strap the opener directly to the enzyme, and both of them work well. We're editing over half of the striatum. That's a region we injected into, and we're editing about 60% of the neurons in the striatum. So this is pretty good. We were curious about using a neuron targeting signal, which is shown here. It looks like it's working kind of okay, but without the opener, the signal isn't very great. However, when we combine both of them, we're able to cover 75% of the striatum and edit 2/3 of the neurons in that area. So this is the best editing we've ever seen in the brain, and it's very encouraging. And we hope that we can leverage the same approach to combine all these pieces and create a self-delivering homing enzyme to find and edit the stem cells in the bone marrow to help with a sickle cell treatment that happens inside the body. So in conclusion, I'd just like to quickly tell you how we want to move from the status quo to an improved administration route for CRISPR. So right now, the patient cells are taken to the lab and an electric shock is applied, and that makes the cell membranes weak enough that the enzyme can slip in. This works, but it requires a clean room and it really limits access in lower resource settings. So what we want to do is improve access to this technology by simply creating these CRISPR cocktails that can be applied to the cells, and you don't need a clean room anymore. You can just do this in flasks, sort of. So this will bring down costs somewhat, and it's going to still require transplant and chemotherapy. So it's going to improve access a little bit, but it's not a real game changer. However, if we can find a way to create this homing enzyme that can reach the bone marrow and edit those stem cells within the body, that's going to be a totally new era in editing, because we won't require the transplant anymore. We won't require the chemotherapy. The therapy can be done in under a week. And we're going to be able to get the cost down to as low as $50,000. And this could even be more affordable as it increases in scale. So with that, I just want to acknowledge everybody at IGI. We actually have our own clinical trial for CRISPR, and that was led by Jennifer and Fyodor, as well as some clinicians at UCLA and UCSF. And really, as a team, we're trying to pull all the pieces together to create an idealized CRISPR therapy. And finally, I want to thank my team who did the work, particularly Maddie and Amir and everybody else who contributed. So with that, I'll be delighted to take any questions.
Thank you very much, Professor Wilson. This is really exciting. Now we have a few minutes for questions. So let's start with the first question. How does this relate to immunotherapies and chemotherapies that are being used for cancer now?
Yeah, that's a wonderful question. So I think CRISPR has massive applications for T-cell-based therapies. So these immuno-oncology approaches that you're talking about, CRISPR has gone into the clinic in multiple contexts, and it has shown promise in that aspect. So it's a little bit of an incremental improvement from the previous versions of anti-cancer cell therapies, but I think the field is really just getting started. So the most exciting thing about what CRISPR can do for anti-cancer cell therapies is that we have so much power to reprogram cells that you can start to create really sophisticated circuits of cells that once they find the cancer cell that they're hunting, they can actually multiply and they can expand in a way that is really, really potent to fight cancer. So I think there's huge capacity for CRISPR to take anti-cancer cell therapies to the next level. And this can be done both outside the body by creating something that's prepared and sitting in a freezer ready to be deployed to a patient. And it can also be used to create anti-cancer cells inside the patient's body. So there's a huge amount of potential there, and a lot of it is already coming to fruition.
That's amazing. So here's a question from the chat from Shrey Patel. Is there an immune reaction in clinics due to off-target reactions or off-target editing? And do these patients need to be put on immunosuppressants after the gene editing to prevent any autoimmune reaction?
Yeah, that's a great question. So I would say one advantage regarding the immune responses is that, with the traditional bone marrow transplant, even when a patient with sickle cell thinks that they have a close relative who's close enough, to have immune compatibility for the transplant, there are some cases where there is still a clash between the patient and the engrafted cells, and it can be fatal. So in this case, that's one fundamental change. Since the patient's own cells are being turned into the curative material, they don't run that risk. And furthermore, the CRISPR enzymes themselves get metabolized very quickly and have a very minimized immune response. So that's another strength. And I would say the off-target aspect doesn't really impact the immune response very much. There's a lot of energy dedicated to making sure that immune responses, a lot of energy dedicated to make sure the off-target edits are well characterized and minimized, but those don't seem to be causing a practical problem so far. So I'm unaware of any time an off-target actually caused the problem. So it looks very safe in that regard. The biggest risk is the chemotherapy that kind of clears out the bone marrow to make the engraftment efficient.
Very, very, very cool. Let's have time for one last question. Do you use in silico, namely AI-based protein design to design the enzymes, namely design it first before you make it in the wet lab, or is everything done by CRISPR editing?
Yeah, that's a great question. So I have not employed much AI so far. However, I'm very inspired and excited to give it a try. So I trained as a structural biologist, and when AlphaFold came online, it was a very deeply personal moment because I saw, what I worked on for a decade just kind of evaporate, right? Like that was my entire career, and now it's just kind of done. So I have massive respect and optimism for what AI can do. And in our particular case, the biggest opportunity is actually finding totally novel little molecules that can recognize the surface of the cell that we care about. And it's possible that the ideal molecule is not out there and will never be generated by an antibody. So using our antibody-based homing approach, that might not be ideal. And I think AI is going to be really adept at looking at the virtual cell surface of one of these stem cells we care about, and then finding all the things that will uniquely bind to that cell. So that is what I think is the greatest opportunity for CRISPR delivery and machine learning. But time will tell. I'm sure there's some other applications I haven't even dreamt of yet.
Thank you. We should now move on to our final speaker this evening, Professor Jeffery Cox. Jeffrey Cox is the CH Lead Chair of Biochemistry and Molecular Endocrinology and Professor of Immunology and Pathogenesis in the Department of Molecular and Cell Biology. The Cox Lab is fascinated by the mechanisms by which pathogenic bacteria manipulate their hosts to cause infections. The lab focuses primarily on tuberculosis, applying a unique perspective on the molecular mechanisms of host pathogen interactions to a broad spectrum of organisms. Over to you, Professor Cox.
Great, thank you very much for that introduction, and thank you everyone for sticking around for my talk. My talk is going to be a little bit different than Danielle and Ross's, in the fact that I'm not representing work done in my lab, but actually 15 labs across both UC Berkeley and Harvard. And also, I'm not going to be showing any data as well. However, we are going to be talking about a novel approach to combat a huge problem in global health, and that is antimicrobial resistance, also shortened as AMR. I first wanted to recognize that this is one of the exciting things about this project is really a new initiative that has just started over the past year here at Berkeley. And it's a true collaboration with, again, 15 different labs, different faculty at both Berkeley and Harvard, in conjunction with a new startup company called Xbiotics to form a consortium where we can attack this huge problem. And this problem is big and important, but also complicated, in that it requires a combination of microbiology, immunology, and chemistry. And I hope that comes shining through by the end of this talk. I think we all, or most of us, appreciate the problem of antimicrobial resistance. It's one of the things that I worry about constantly, and that is that the specter that we will run out of antibiotics that work, and that we'll be infected with microbes, including bacteria, that are resistant to what we use all the time, and that's antibiotics. And this is data that was generated from a couple of years ago, 2019, just documenting the different ways that humans die over the globe. And you can see that there's a lot of infectious diseases. Cancer is a huge one. And up here, the AMR now is that today about 1.2 million people die every year from infections that are untreatable with antibiotics. This, of course, is a big problem, but an even bigger problem is that by the 2050, it's estimated that this number is going to rise dramatically, 10 million people a year, and even become bigger than cancer eventually as a mortality causer in humans. So this prediction is that we're in a tough spot, and we have to do something. We have to do something different than what we've been doing over the past few decades of antibiotic resistance and new antibiotics. The real problem is that resistance is outpacing discovery of new molecules. We know that if we have a bacteria here on the right that can infect humans, is that in most cases still, or in many cases, that there are antibiotics that can work and that they can kill the microbe during infection of humans, and then it can clear the infection. However, what can happen, of course, is that these bacteria can mutate and become resistant to these particular antibiotics. So then to treat a subsequent infection, we then have to come up with a whole new antibiotic that will kill this same microbe. And so, again, the problem is that we're just basically chasing our tail, and that we're going around where there's generation of antibiotic resistance, and we have to make new antibiotics that will now kill those microbes. Our idea is to create resistant-proof antibiotics. Now, that it's easy to say, but it's hard to do. And our idea is that instead of keeping on with this vicious cycle of trying to find new antibiotics that kill the microbe, is that we want to target the host to activate natural microbe killing mechanisms that are inherent in us. And this is what merges the microbiology, immunology, and chemistry to come up with a new way of thinking about antibiotics that break this cycle of resistance. In order to understand what we're trying to do here and how to make an antibiotic that is immune to resistance, it's important to understand how resistance comes about. Of course, we can get infected with an already antibiotic resistant microbe, but really how the evolution of antimicrobial resistance comes about is during clinical use of antibiotics. And so on the left here, there's a pool of bacteria, that most of which are sensitive to antibiotics. But because of background mutagenesis rate, some of those bacteria in this population will actually have resistance to the antibiotic that we'll use. So if we take that population and now treat them with the antibiotic, of course, all the sensitive ones die, but it's the resistant ones that are still there and not only alive, but also to reproduce in the presence of the antibiotic. And this leads to antimicrobial resistant infections. And so it's really that evolution that we're trying to block. How do we make an antibiotic where these small populations of bacteria are actually killed? So in order to create a host-directed therapy, we decided to look primarily at macrophages. And macrophages are host cells in all of us that are important for controlling lots of different infections, including bacterial infections. And these are cells that patrol our tissues. And when they come across say, bacteria here in light blue, is that they can engulf the bacteria and they're able to kill these microbes. So that's the natural killing mechanism. It turns out that in the immune response is that there are activators, there are gas pedals on the immune system. And one of those is a cytokine called interferon gamma. And by expressing interferon gamma, now these macrophages can respond to this cytokine to become even better at killing microbes. And so therefore, it leads to greater inflammation and a greater antibacterial state. Macrophages, however, are complex cells. They are not only involved in killing of microbes, but they're also important for the opposite effect, and that is to actually now restore the tissue after an infection or a wound. And so they are also have functions that are anti-inflammatory and help promote wound healing. And this is directly in contrast to this activated inflammatory state. So they can be both inflammatory and anti-inflammatory. What we've learned over the past several years is that under the conditions of many different bacterial infections is that this dichotomy of macrophages, that their ability to actually be anti-inflammatory can actually promote a situation where the bacteria can grow. So these macrophages are under the control of a bunch of different cytokines. And if the wrong cytokine is made, for example, anti-inflammatory cytokines that can actually counteract the killing activity of interferon gamma. So you can see that macrophage activation is under multiple modes of regulation. Anti-inflammatory cytokines can dampen macrophage activation, which leads to increased susceptibility to infection. But pro-inflammatory cytokines can boost this antibacterial effect, but also too much can actually lead to uncontrolled inflammation and autoimmunity. We think that one of the problems during infection is that this balance is skewed too far to the anti-inflammatory. So our solution is simple, is just add more interferon gamma to tip the balance. That is to add more pro-inflammatory cytokines to now make these macrophages better at killing these microbes that are difficult to kill with antibiotics. The problem, of course, is that systemic interferon gamma is toxic. In fact, I'm sure we all remember from our COVID days, this idea of cytokine storm, where stimulators of the immune system can actually now lead to these feed-forward loops where we actually are inducing so much inflammation that that actually causes tissue damage and severe disease in the human. So how do we get around this problem? Well, this is where the chemistry comes in, and that is that our approach is to use these molecules, these bifunctional molecules, and they're termed immune-linked antibacterial chimeras, or ILACs. And the two functional parts of it are at the top here, and the purple is a cytokine, for example, interferon gamma. And on the bottom here is the bacterial binding domain. And these are held together by a linker. So they're bifunctional in that these will bind to the outside of a bacteria, but now also deliver the cytokine specifically to the site of infection. And so that's diagrammed here. And that is these green bacteria have an ILAC bound to them, but in the tissue, there's also macrophages that are moving around. And this will now deliver interferon gamma specifically to these macrophages in vivo and will lead to a zone of killing. Okay, so this is my last slide. And it's just to get back to this idea of why then would ILACs circumvent resistance? And the basic idea is that not all bacteria need to bind the ILAC. In the case of antibiotics, they all have to be sensitive to the antibiotic, but in this case, it doesn't have to be. For example, if we had an ILAC resistant mutant in this population, and now we added an ILAC, and say this one bacteria, this red bacteria is still resistant or it doesn't bind to the ILAC, it still is sensitive to the recruitment of the macrophages in that site, and that it will also be killed just like the sensitive ones. So that's the story. That's our idea, and I'm out of time. And thank you very much for your time. And I'm happy to take any questions.
Sorry, it works better if I unmute. Your lab is doing really noble work, and we have time for a few questions. One question from the chat from Susan Aquistapes is your group exploring the use of phages targeted for each pathogen?
Yes, I love that idea. We, in fact, phages have the same idea, right? Is that they can bind specifically to microbes. And one of our ideas, although we're not doing this yet, is that in that blue thing that binds to the bacteria, if we could use phage proteins to attack that, I think that would be great. However, in another, in my lab, we are using or attempting to use phages as a way to attack mycobacterial infections. And it turns out that there is a unique interaction between antibiotics and phages. And this is new work, not from our lab. And we're trying to harness that this evolutionary relationship between phages and antibiotics where they can synergize in vivo. In fact, I'm actually teaching a lab class next year where we're going to be exactly of this for undergrads. But, well, they'll be actually doing that work in identifying the phages that synergize with antibiotics and mycobacteria. So yeah, I'm very excited about phages for sure.
Oh, thank you very much, Professor Cox. Now, all of these presentations have been fascinating, and with the little bit of time we have left, I would like to invite all of our speakers back for some last questions. And as people think about their last questions, I would like to ask Professor Cox one more question. Given the delicate balance in the immune system between too little when the infection wins and too much where the immune system overwins, when you're working with the ILACs, there must be such a thing as too many. How do you find the right balance?
Yeah, no, that's an excellent question. First of all, the biggest issue or the best aspect of these ILACs is the fact that we hope that they concentrate at the site of infection. So that way, all of those nasty reactions are happening at the site of infection where they should be and not out in other parts, uninfected parts of our tissues. The other thing that we can do, though, based on what we know about these cytokines, we can actually tune the activity of these cytokines such that we can make them more potent or actually make them less potent. So there's a lot of play in there to have a little rheostat for the amount of inflammation that we can obtain from these ILACs.
Thanks. So I see that there were more questions in the chat to our previous speakers. But I also see with delight that many of those questions have been answered also in the chat. So at this point, I would like to start to wrap up our very interesting hour and thank all three of our speakers for your fascinating talks. And I'm going to hand the floor to close to Assistant Dean Maria Helm for her closing remarks.
Hi there. Thank you so much, Professor Jacak, and to Danielle and Jeff and Ross. I'm really grateful that you all came out this evening and grateful to everyone in our audience. This was an incredibly interesting conversation. So as Assistant Dean, and I'm Assistant Dean of the Division of Mathematical and Physical Sciences, one of the aspects I love about Berkeley is that I'm always surrounded by super smart people like Danielle, Ross, Jeff, and Barbara, and super smart people who are doing groundbreaking work and making great discoveries. And this is what basic science is about. I hope that with these events and that tonight we've brought this feeling to all of you, no matter where you are. The university's commitment to research excellence and innovation ensures that it will continue to be a leader in scientific discovery for years to come. This is what Berkeley's known for, after all. So we have a couple more of these basic science events in the coming weeks. Next week on April 30th, we will have a really wonderful presentation on the science of teaching. So that will be 3 scientists who are renowned for their teaching skill, and all of them have actually won the Distinguished Teaching Award at Berkeley. So they'll be introducing their science, but also talking about how they engage students in scientific inquiry. So I think that will be a special one and super interesting. Please reach out to us, to me, to anyone, if you would like to know more about anything we've covered. We'll be sharing some additional resources, and in about 2 weeks' time, this video will appear on the Basic Science Lights the Way website. And that website is just chock full after several years now of amazing videos of the events we've put on. So I encourage you to go and look at past events and past years. With that, I do want to acknowledge that we scheduled this event on the very first day of Passover, and I'm sorry to anyone who missed it for that reason. But I do want to say Hag Sameah and happy Passover, and stay curious, Fiat Lux, and go Bears. Thank you so much for all your support and for attending tonight.